this competition. And 7 of those awards came to BC. And I mean, as you know, we have about 13% of the population. So we did way above what we were really projected this on the basis of population. And that just speaks to the strength of the bioinformatics community that we have here in BC. So we ended up achieving about $6.2 million that was awarded nationally, about 2.7 million. So we're very pleased. And there were 4 large-scale projects that were awarded and 3 smaller-scale projects. So the 3 larger-scale projects are listed here. All the projects are listed on our website. And what we're going to do today, I'm not going to really get into detailed introductions because you all know our people from our bioinformatics community. And it's starting with Wyeth Wasserman from the Center for Molecular and Medical Therapeutics. And what we're going to do is just each of the speakers is going to have 10 minutes. And it's really to share with you the excitement about these projects and what these groups are embarking on. So with that, I'll just turn it to Wyeth, who's going to talk about the applied bioinformatics of cis-regulation for disease exploration. So please join me in welcoming Wyeth.
Thank you all. A little echoey, we'll see how it goes. It's wonderful to see lots of familiar faces here, and it's a tremendous feeling within the bioinformatics community that we've done so well overall, and so I'm very pleased to be kicking off this section. It's going to be very different than what you had last time because you're going to get this machine gun style for the next few minutes. So, protein coding regions of genomes have been available now for a while, and our group and many groups have been participating in sequencing exomes and using those to discover disease-causing mutations, and it's been tremendously successful. So this is just an example of one coming out of BC Children's Hospital in the TIDE project where we look at metabolic disorders. And this is a case where we mapped out a homozygous recessive mutation in the CA5A gene, which affects a key portion of the metabolic base of the enzyme. It passes from heterozygous parents to affected children, and in this project we were able to identify the mutations and determine the newborn baby was not at risk for the metabolic problems. So it was a real success story. But my own passion lies outside the protein coding regions, and so I'm much more interested in moving out of the exomes into the whole genomes. And so about 5 or 6 years ago, Jared Roach from the ISB sent me this slide, which lost our stat around the edge, but complete genome sequence stat, and I thought, okay, that's a long way off. It's going to be a while until our clinicians are calling down to the halls and saying we need a genome sequence and better get the clinical bioinformatics expert into the operating room so we can do something. But actually, something almost like this happened to us a few months ago, and so we had a case at BC Children's Hospital where there was a phenotype of the individual. The treatment of that individual was dependent on finding mutations in both copies of both alleles of the gene that's known to be relevant to the treatment. Treatment costs only like $250,000, so there's no go unless you confirm that it's caused by this particular gene. Sequencing of the exons identified one mutation in one allele, but not in the other, and so we were able to send it off for genome sequencing to try to identify whether or not this was a suitable case to take forward. In the end, we actually found another mutation in another gene that was responsible, so in many ways it worked, because we didn't give them a wasteful treatment that was going to benefit them. But it was the first case where the clinical genome stat actually was relevant. So here's your genome. You've got a bunch of exons in it. You've seen these types of pictures before. But what I'll assert to you is that outside of those exons, there's another whole world of interesting features in the genome that are regulatory. And regulatory in many ways. They can be transcription factor binding sites, they can be microRNA target sequences, they can be a variety of long non-coding RNA sequences that are involved in the regulation of genes. A bunch of interesting regulatory sequences. And if we pull those apart and we sort of start lining them up, we see, okay, there's a protein coding space, there's a regulatory space, and then there's a big space that we don't really know very much about. But over time and over the last few years, what's happened is that big black line is getting shorter and shorter, and what happens as we go along is that eventually it's all regulatory, because this is the key issue of understanding our genome and understanding how the rest of the regions affect which of these exons are active at any given moment. So the project that we got funded to take forward, ABC4DE, is focused on how can we develop informatics tools that allow us to analyze and interpret genetic variations that are falling within these regulatory sequences. So can we give a creation a toolbox and a toolkit and a methodology that is useful and relevant to get people's attention to mutations in these key regions. So loosely, I'm just going to give you the vaporware version here, which is sort of 3 steps in the process. The first step is essentially defining where within the genome we think those regulatory regions are located. And we build off of things like the ENCODE project and work that's going on in labs all around the world who are trying to develop methods and tools for calling where these blue regions are, these regulatory regions are in the genome. Once we've identified those regions that are regulatory, then the key thing is to start going in and looking to see what mutations might be functionally consequential in those zones. And so I'll give you an example out of my passion, which is transcription factor binding sites, but there are a variety of tools and methods that you can take forward besides that. So within the world of transcription factor binding sites, we will use data sets like ChIP-seq data sets, chromatin immunoprecipitation. And what you can see here is an example of an OCT4 transcription factor study. And you can see within the ChIP-seq data, a subset of the data has the binding sites for that factor. The other are coming along for other reasons. But we can now computationally take the experimental first pass, then narrow in on those ones that have the correct motif for the transcription factor to stick to. And then the final step, which is to say, once we've identified where there's an element that we have high confidence in, can we identify that there's a variation that's overlapping or present within that, and that variation might have an impact on the regulatory event. And so that goes to the bread and butter of informatics, which are tools like motif discovery, motif analysis tools, where you say, here's a motif model. We can look at 2 different alleles, score the genome for the impact of the mutation on the predicted interaction of the protein with the DNA. And then we can look to see within the distribution of possible impacts, does that variation look like it's falling outside of the norm and is going to be an extreme impact on the function. So we've implemented the pipeline, and we've been working with our team to take it forward on the first sets of genomes that we can get our hands on, and we're now going to, for the next 3 years, be refining it, putting the tools out there for everybody to come forward and work with, and hopefully collaborating with all of you in the community as you get your whole genome sequences coming back to see if the tools are working well and see if we can facilitate your work in finding regulatory changes that are affecting gene expression. So the team that's coming together is 4 groups that are involved. So Anna Lehman is a clinical geneticist at BC Children's Hospital, and she's helping us work on sort of the clinical interpretation of regulatory variants. So this is a challenging issue of saying, does this variation actually have relevance to the clinic? Does it mean something? What's sufficiently interesting that you can take it forward and do something in a clinical case? Claire von Karnevek is working with us on regulatory variants in pediatric metabolic disorders. So she's one of the leaders of the TIDE project. They're doing genome sequencing, and we're looking for changes that affect metabolism in those cases. With Saurabh Shah, we are looking at cancer genomes, have our first sort of analysis set that's working its way through right now, looking at regulatory variants in cancer genome sequencing. And then my lab, which is developing the software and methodology. So in rapid fire fashion, thank you very much for your time. I think we're taking questions at the very end of the set, and I believe